Now we are discussing about physiology of muscle contraction. Muscle contraction include three types of muscles: muscle tissues, skeletal muscles, cardiac muscles, and smooth muscles. Smooth muscles tissues are non-stretched, involuntary, fusiform cells, one nucleus per cell, wall of hollow organ that is stomach, walls of tubes and passageways that is broncholysis, blood vessels, esophagus. Slow wave-like contraction. Smooth muscles. Description is that spindle-shaped cells with with central nuclei, cells arranged closely to form sheets, non-stations, functions, prop, propels substances or objects, foodstuffs, urine, a baby, along internal passageways, involuntary control. Location mostly in the walls of hollow organs. This is the diagram and example of the smooth muscles. In the microscope, we can see that this like of smooth muscle cell and nuclei, cardiac muscle tissue, straight involuntary, fit together with tight junctions that promote fast conduction of electrical signals. Heart branching cells, one or two nuclei per cell, medium speed contractions. This is the cardiac muscles of diagram and example. Description: branching straight, generally uninucleate cells that integrated at specialized junction, intercalated disc function as it contracts, it pupils blood into the circulation. Involuntary control also. Location: the walls of the heart. In the microscope, we can see that the intercalated disc and nucleus of the cardiac muscles. Skeletal muscles tissues are straight, voluntary, attached to bone via tendons, fascia or skin. Long cylindrical cells, many nuclei per cell, rapid contractions. Skeletal muscles, long, cylindrical, multinucleated cell, obvious straightness, functions, voluntary movements, locomotions, manipulation of the environment, facial expression, voluntary control. Location: in skeletal muscles attached to bone or occasionally to the skin. In the microscope, we can see that of skeletal muscle in this nuclei and part of muscles fibers. Function of skeletal muscles is that produce heat, produce movements, maintenance, body posture, assist in joint stability. Contraction of skeletal muscles. The skeletal muscles is contractile in natural, that is, it contracts when excited. Excitation's depolarization is called also depolarization of muscles fibers causes development of action potential which through sequence of events produce muscle contraction. These two phenomena are coupled together by calcium ion. Thus, the process by which depolarization of muscle fiber initiate contractions is called excitation control coupling. It is the contraction of muscles fibers, actin and myosin. Actin thin and light, myosin is thick and dark, arranged in bands that slide past each other. Calcium and adenosine triphosphate are required in actin and myosin also. Actin is thin and light, myosin is thick and dark. Please do not confuse in the actin and myosin arranged in bands that slide past of each other. Calcium and adenosine triphosphate are required. Excitation contraction, commonly called EC coupling. This is the diagram you can see in a right direction. First one, action potential generated and propagated along sarcomere to T tubules. Action potential triggered calcium release. Calcium binds to troponin, blocking action of tropomyosin released. Contraction via cross bridge formation, ATP hydrolysis. Removal of calcium by active transport. Tropomyosin blockage restored. Contraction ends. This is the process of excitation contracts coupling. We are again repeat. Action potential generated and propagated along sarcomere to T tubules. Action potential triggers calcium release. After the calcium release, calcium bind to troponin, then blocking action of tropomyosin release. Contraction via cross bridge formation, ATP hydrolyze. Removal of calcium by active transport. Tropomyosin blockage restored and contraction again ends. Muscles protein. In this first graphic, the myosin binding sites on the actin proteins are readily visible. You can see in the myosin binding sites in the diagram, the regularity proteins troponin and tropomyosin have been added to the bottom graphic. The myosin binding site have been covered. The regulatory protein troponin and tropomyosin have been added in the bottom graphic. The myosin binding sites have been covered. This is the diagram of the myosin binding site. Electrical phenomena and ionic fluxes. The electrical events in the skeletal muscle and the ionic fluxes is quite similar to that in nerve with quantitative difference in timing and magnitude. The RMP of skeletal muscles is about minus 19 MV megawatt, 90 megawatt minus 90 megawatt of the RPM of skeletal muscle. The AP, it means action potential. The AP lasts 2 to 4 muscles and is conducted along the muscle fiber and about 5 minutes per second. The absolute refractory period is 1 to 3 minutes per second long, and after polarization are relatively prolonged. Ionic distribution and fluxes. The distribution of ion across the muscles fibers membrane is similar to that across the nerve cell membrane. Approximate values of the various ion and their equilibrium potential are shown in table. As in nerves, depolarization is largely manifestations of the sodium influx, and repolarization is largely manifestations of potassium influx. Now, contractile response. First one is that electrical events, mechanical events. Although one response does not normally occur without each other, but their physiologic basis and characteristics are different. Then muscles fibers membrane depolarization starts at the motor nerve plate. The action potential is transmitted along the muscles fiber and initiates a contractile response. Molecular basic contractile sliding filament theory. It is a very important theory in your syllabus. Please do not avoid this theory. आपके एग्जाम में भी ये पूछी जा सकती है, तो आप इसे अच्छे से पढ़ लीजिएगा. The process by which muscle contractions occurs in brought about by sliding of the thin filament over the thick filaments. Okay, I repeat, the process by which muscle contraction occurs in brought about by sliding of the thin filaments over the thick filaments. This shortening is not due to actual length, but it is brought about by increased overlap of the thin acting and thick filament myosin with the muscles thin. I repeat, this shortening is not due to actual length, but it is brought about by increased overlap of the thin means actin and thick filaments mean myosin with the muscles thin. Thank you.